So here we are again, I think. Hi, hello everyone. Okay, everything seems to be working. Hi, hello, and welcome to another Saturday microscopy live stream. Maybe in some places of the world it's already Sunday, Sunday early in the morning. Hi. Yeah, today, look, I'm going to put these are worms or were worms in here, and it didn't work. The culture really got moldy, so we're going to have a closer look here today at those nematode worms that I've been growing here more or less successfully. Yeah, I also have today, yeah, in case we run out of time, some other mold and bacteria from some food, from cheese, that I would like to observe together with you. Yeah, and I would like to usually to spend the first couple of minutes, like always. I would like to just say hello around the world. So I usually read out, yeah, some comments. So if you are joining in here, just please feel free to also tell the community where you're from. Okay. Usually I read out here. Hello from Oregon in the United States, from New Jersey, from northern France. Yeah, from Georgia, from Tennessee in the United States, from Florida, Vietnam, yeah, Athens in Greece, England as always, and a very nice comment here. Hi, Oliver. I hope you realize that this is the best thing on TV on a Saturday. Honestly, I mean, I'm a little bit speechless. Thank you very much. Hi from Poland. Yes, happy Sunday morning from. Where is that? Okay. Heads up, your email inbox is full again. Gee, sorry about that. Okay, thank you. Um, that's interesting because I usually try to clear the email inbox. Okay, yeah. So in any case, from Styria, from Austria. Okay, not so far away from where I am, because I am also basically broadcasting from Austria in Central Europe. Yeah, and today I would like to do the following. Look, yeah, I know every now and then I put some nematode worms under the microscope, and I'm kind of culturing them. I have been culturing them now for about 3 years. And this is the jar that was successful, and we're just going to put them under the microscope as well. They're known as vinegar eels, and they're sold as fish food. And people who have a home aquarium, they kind of grow them, and they move very quickly. So I would like to slow them down using some methyl cellulose, so we can really zoom in and look at them. But there is a problem that I have, and that is that essentially two of my cultures died for two different reasons. I don't know. This one over here, you can see that this is weird stuff floating around in the culture here, and these seem to be some kind of fungi or bacteria. But the worms, they completely disappeared, so they died off. So we're going to, and I have a suspicion of why this could have been. So we're going to put this a little bit under the microscope as well. Maybe we're able to find out what this stuff is that's floating around here. Suppose maybe some fungi or bacteria. And this one here is pretty bad. Look at this. This one actually got moldy. And I have, however, seen that if you go around here, and I don't know that you, yeah, actually maybe you're able to see this a little bit. If you see those tiny things floating around in here, yeah, these are actually the nematode worms. It's not in focus, unfortunately, because I cannot focus that closely. But they have somehow survived in here. Yeah, so you see that there are tiny things swimming around here. But yeah, the surface over here is all moldy. And this is now interesting because they have survived, and I think the reason is because even though there is mold on the surface, oxygen still could diffuse through into the vinegar, because this is actually a mixture of vinegar and sugar. And this one over here, I suppose that these might have been bacteria, and maybe they kind of formed such a dense layer that oxygen was not able to go into the jar. Right, so this one here actually, yeah, is not a nice one, and this is the way that this one is intact. Okay, so I'll be using this one here, and you see how dense it is, almost impossible to see now, right? And I've included down here this white stuff that you see down here is or was a piece of apple as well for additional nutrients. Yeah. So I want to basically subcultivate them. So I want to take out a sample here and I want to put them over into another jar. And then after a couple of days or weeks, I hope that they're basically also growing, because I'm kind of worried that this jar over here also, maybe the culture is going to die off after some time. So that's what I'll be doing today. And then later on, because school has started a few weeks ago, I basically have started teaching biology lessons again, and I was talking about different cells and so on. And of course, also mentioned that, biotechnology and cheese. I really love cheese, right? And I told my students, of course, there are also cells growing on cheese. And some of the students, they were completely surprised by that essentially, we're using microorganisms also as a food. And this is actually a recommended way of using specimens from a safe source, because sometimes in schools and at home you want to look at some bacteria maybe or some fungi, and you don't want to spoil food because that might not be healthy. But if you use the fungus and the bacteria that are growing on cheese, I mean, then these are known species, known cultures, and they're a safe source. So why am I saying this? Because every now and then I read online, I want to observe some bacteria and should I spoil something, and then I can see bacteria and I'm saying, no, don't do that. It's not necessary. Just take natural bacteria that can be found on food. Okay, so that's simply my general recommendation. Yeah, if you have any questions or any comments or so, please feel free to post it in the how do you call this, in the chat. And another thing that I have is that I want to show you, and this one over here is a couple of weeks ago I received 2 jars with water fleas Daphnia. They did not quite survive very well, even though I fed them nicely. But in this one jar, and last week I also showed you, difficult, it doesn't focus again, but these are oligoheat worms. These are segmented worms. And they are Iolosoma, one of my favorite ones actually. They're much significantly larger, and they look pretty nice and cute. So I also would like to, I have not tried this either yet, I also would like to try to observe them using methyl cellulose to slow them down. So that's something that I don't remember that I have ever done with those worms here. So usually in these live streams I usually experiment around, and I usually try to observe things that I have not yet tried out in the same way before. So what we're going to be doing right now is we're just going to have a look at some of those worms. I know many of you have already seen them before, because they're one of those specimens that I always return to. But specifically, I would like to have a sample here from the surface, the fungus that's growing on here, the contaminant. And that's and I'm going to close the other jar here, just to make sure. And can I make permanent slides with fungus? Of course you can. Of course, there are different ways how you can do that, and if you want to, you basically in a mounting medium, usually I use some glue to do that, or some other mounting medium. You just make sure that the fungus is it depends a little bit how you want to prepare it, which fungus it is. Maybe that's also something for next video, the preparing of not mold, which is a fungus, but actually, edible fungi. That we use as food, how to slice them maybe, and how to prepare them. So let's have a look at this one over here, because this is, again, this is something I did not observe yet. I use those pipettes, as always. I go in here, I go to the surface, and I want to take a small sample from the surface here. And let's have a look on what we're actually able to see here. Cover glass goes on top, as always. And again, this is on, that's a little bit part of the format of this live stream, that some of the things, or many of the things that I'm actually doing here, I'm doing for the first time. So there is also a high element of surprise involved. And it can, of course, be that the specimens that we see are completely boring and not interesting at all. Or we discover something unexpected. So that's a little bit, part of this format. I put this on here now. Let's have a look at what we're able to see. I hope that I caught also some of those nematodes. And the scope view, it always takes me a little bit of time to change the how do you say? Yeah. Okay, yeah, let's flip this out here. The rainbow colors is because of the polarizing optics that I have. So let's do it like this, yeah. And yeah, we can of course see that yeah, those worms are present. Then there's this. I think I have to zoom in a little bit more. Again, those worms are completely harmless, and they are sold as fish food. And what you see here, and that's I need to switch on the arrow, and I think that's the stuff that you see here in the background. This must be somehow the fungus, the contaminant. Yeah, and maybe they feed on that. I don't know, right? So let's go in here. But in any case, I don't want to have that fungus. Yeah, in there. I'm going to just put in this filter again here. Yeah. And yeah, these threads, yeah, indeed appear like a fungus. Yeah, it's very clear. I just want to explain to you. If you look at this one over here, for example, you actually see that it is branched. Yeah, there's a main cells over here, and then it branches off and goes to the side. And now you see this. And if it's branched, then this is a very, very strong indicator that we're actually looking at fungi and not bacteria. Why am I saying that? This was actually one of the examination questions that I had to pass at the university. It was an oral exam, and they basically, the professor asked me, how can you distinguish under the microscope, how can you distinguish bacteria from fungi? And yeah, and one of the, of course, fungi are significantly larger. Yeah, but one of the factors is that there is branching going on. Yeah, and yeah, you can this is a so-called a septate fungus. That means you can see the individual cells here. Yeah, and then sometimes it branches off. Yeah, like over here, for example. Yeah, so that's basically, obviously, I'm kind of confirming under the microscope what we already knew, that it's basically a fungus here. Yeah, yeah. So that's a little bit how it looks like, and yeah, let's go around a little bit here. What about spores? How to make them in a permanent slide? Honestly, not a. You're asking. Thank you for the. You see, I want to reward people who are asking questions. Then what I'm going to do? Then I'm going to slightly modify later on, if I have time. I'm going to show you how to prepare spores. And I need to get the mounting medium, which I've got somewhere in my cupboard up there. But I've got some spores here. Okay, which you cannot see now because of it's too bright. And yeah, I'll show you how to do that. Yeah. So this is not a problem. Actually, fairly easy to do. And yeah, that's basically something I don't want. Like over here, I mean, these fungi. What I have, however, also once observed, and this really requires a lot of patience. You can sometimes actually also see the fungus grow under the microscope. Yeah, so you can actually see how those the hyphae or hyphae, depending on how you want to pronounce it, how they can actually grow. And in time lapse, that's also possible to see. Yeah, of course, not enough time to do that now. Yeah, but again, yeah, that's the contaminated sample, and here are the worms. Yeah. I have a camera question. I use a Canon camera using a 2x adapter through one of my binocular eye spaces. I want to know if I do better if I get a Swift 5 mega. Oh, this is a complicated question because it does not only depend on the camera itself. It depends so much on the complete setup, and connecting a camera to a microscope is a science. And what I found out is the following. I found out that trial and error is probably the best way. I've seen so many factors and parameters influence the quality, and generally, if you're using a Canon camera and if you're recording directly into the camera, it also has an advantage because it's much faster this way, especially if you want to make videos. And if you want to connect a camera directly over USB to the computer, then sometimes you do not get a very high frame rate. So even that might be a factor, right? So that's a little bit a thing. However, if you get a camera, you just basically plug it into the trinocular head of the microscope or into an eyepiece. So generally, that is much more easier to use, right? but honestly, every camera setup has its own advantages and disadvantages. Yeah. So I agree with ever smaller. If you get yourself a microscope camera direct connected to USB, it's pretty much plug and play. Yeah. But some cameras need a special camera software. In any case, before I talk too much, that's basically how it looks like. And the worms move slowly because, and that's maybe a good point here, because they're surrounded by the fungus here and because it's kind of compressed, right? So this limits the movement of the worms, right? I'm going to show you now the other sample. Where the worms move much faster, and then we have to slow them down using the methyl cellulose. Okay, so that's a little bit the thing I want to do now. I'm going to go back to the desk view. I'm just going to show you the spores. Okay, so these are spores from a fungus that I've isolated from a rotting lemon. So what I've done is I put some squeezed out lemon juice on some filter paper, coffee filter paper, like the one that you use for making coffee. And then I added a couple of drops of lemon juice, and then I kind of put some of the spores on here. And after a couple of days, this is how it looked like. Not a good idea to open it up. It's not healthy, but if you want to use look at the spores of a mushroom that you can eat, what you do is you just take the cap of the mushroom and you put it on a slide or on paper, and you just knock the back of the mushroom. I'm just gonna if this is kind of the I don't know what to take let's use this here. If this is kind of a mushroom, the head of the mushroom, and you have got the lamellae with the spore-producing cells on the bottom, right? And you put the mushroom here, and then you take something and you knock on the mushroom, and then you will have the spores being knocked out. And if you have a microscope slide there, then you can collect the spores. Yeah, or you can do it like this. You can grow them. Yeah. So, but I would like to now show you, as promised. How do you? I need another slide here. By the way, some people are asking, do I throw them away, those slides, or do I reuse them? I always wash them and reuse them. So, and that's also important. I always have to exchange. Look, I always have to exchange the tips, because I just went into the jar with the fungus, and I do not want to contaminate the clean jar. So I'm going to just go in here. That's the one with the, and just going to take a sample in here. And let's put another cover glass on top. Let's see, I have to make sure that there's only one of them, because sometimes they stick together. And let's have a look at this, and in the hope that there are also some nematode worms in here. Of course, there will be, but sometimes I'm able to catch more, sometimes less. So I need to always change over to the. Here's one, and you see they move much faster because they're not inhibited by any fungi or so. Let's go down with the magnification a bit further. Yeah, it will. Yeah, so that's how. Yeah, and they move fairly quickly, right? So I need to slow them down a little bit. But as we enjoy them wiggling around, let's have a look at a few more questions. Off topic, it would be fun to see a dedicated video or stream of all of the camera setups and the parameters that can influence the image quality. That is actually a good point, and I've been thinking about that. Maybe I'll talk a little bit about this later. The only problem that I have with that, and it's a technical issue, I'd have to set up a second microscope and kind of show it. I don't know how to do that. So that, because the current system that I have is now quite optimized, but maybe, I don't know, I can do a dry run and can simply show you all of the equipment that I've been through. That I could do in any case. Yeah. Okay. So yes, okay. No, thank you for the recommendation, and why not? Yeah. Do you have any idea on what type of fungus this might be? It would be fungus specific for vinegar. It doesn't have to be specific for vinegar. It's got to be acid tolerant. And honestly, I don't know. And the biodiversity is so extreme that you'd probably have to do a DNA test. Okay, so that is simply something, yeah. Yeah, I do have indeed several videos. If you go into this channel right now, there are indeed several videos that I've made in the past, but I do get it. Maybe it would be nice to have again an updated version of this. So that's basically what I would like to do now is I want to really slow them down. So what I'm going to do is the following, is I'm going to take another slide. Okay. And by the way, it's also possible to use a mobile phone as a webcam. If you install the appropriate software, then that's also possible. Yeah. So even if you don't have if you don't have a trinocular microscope, you get a mobile phone adapter. It's also possible, not quite as stable, not quite as convenient, but also kind of works. Yeah. So let's have again a look at the desk. And what I'm going to do now is the following. Again, I made several months ago or last year, I made a video on methyl cellulose. So I'm taking again this one from the good sample. I'm going to put the other one here. I'm going to put away because I don't want, you see, I'm kind of always worried that if I kind of bump into it, then the whole thing falls over. Not a good idea. So let's put this on the side somewhere. And maybe I might be actually dumping it once, I was able to. Yeah, and later on we also have to put this one here under the microscope because the worms here started to grow and then all of them died. So pretty sad. So, and I have to throw away the tip again. And I'm now going to take a little bit of methyl cellulose. So what is this? This is methyl cellulose is a powder that you can buy. I think I bought mine on Amazon. And you need a very small amount, and you mix it with water, and then it dissolves. Very difficult at the beginning, and then after a day or so, it becomes very clear and thick. And then what you do is you take a little bit of this stuff. It's almost like honey a little bit. I think maybe this is enough. I don't know. And you put it in here. And you don't see it. Here we go. Yeah. And you mix it by drawing it in and up, and down. And this makes everything quite viscous. Now I do hope, and I don't know, because of the vinegar, and you see there are some bubbles here as well. Let's put this away again. And then, of course, another, cover glass. We just ignore the bubbles. Usually they are pushed out to the side after some, And then any worms that are in here should be actually moving much slower. This is actually also a very good, technique to be used for other fast-moving microorganisms. And where even am I here again? You see, I always have to, Yes, here we go. You see those big air bubbles? Yeah. And the nice thing about, and the worms now move much more slowly. And the air bubbles, we just ignore them. And the good thing about methyl cellulose is it does not seem to be osmotically very active. This means that the organisms they don't dehydrate or lose water because of that. Yeah. So exactly. Jess Eversmall is saying it works great to see the beating cilia of the hypotrix. So there are basically, if you have beating cilia, and I actually hope. I have to be honest with you, that's actually one of the things that I hope to be able to see with the Iolosoma worms, because they also have cilia on their head, and maybe I'm also able to see those, right? And depending on how much methyl cellulose you mix into the liquid, basically this is how fast they're moving. Here we go. Yeah. So they're still moving fairly quickly, but much slower than before, obviously. Yeah, so I might have to try to add a little bit more methyl cellulose. Yeah, but this is actually a really nice thing, and which I can recommend. So methyl cellulose is a white powder that is used also for cooking. So if you want to for example, make food a little bit thicker. I also tried in the past starch. So I added some potato starch or yeah, and I tried with water, and then I boiled it to make a thick thing, syrupy thing. But this potato starch didn't work well because you could still see the individual starch grains. They started to swell up, and it was not homogeneous. Yeah, so it didn't work well, but this methyl cellulose is completely homogeneous. It's completely uniform, and yeah, and quite nicely visible. Yeah, the movement is different. Probably fighting the stuff that could be. I don't know if the thing is the following. Interestingly, it doesn't seem to hurt them so much, right? Yeah. So yeah, is it? No, this is not phase contrast. This is DIC. But if you basically would like to see it in phase contrast, why not use this now as a, how do you say, yeah, simply to show you also the different contrasting techniques. Yeah, for those of you who have not seen it yet. So this one is DIC. And look what I can do. I can do all of these fancy things here by changing, yeah, by turning something here. Yeah, I can change the colors. Yeah, so that's the reason why I like DIC, because it's able to add a little bit of color to those YouTube videos. Yeah, yeah. Do they also grow by new heads by themselves? I don't think so. Those nematode worms, they actually should reproduce over eggs that they lay. I've not seen them in asexual reproduction, but this does not mean that they don't do that, okay? But the other ones, the Ilosoma worms, which I'm going to show, they actually will reproduce mainly asexually, and they will actually grow new heads right in the middle of the body, right? So that is, yeah, that's DIC. Most of you are going to see it in bright field. So that's too much light, obviously. So this is regular bright field, okay? So these are the natural colors. Yeah, so you see DIC where you are adding the color, that's not, yeah, I mean, looks nice, obviously, but I mean, it's actually strictly speaking artifact, right? So that's a regular bright field. And you asked about phase contrast. Generally, phase contrast is much better for very small and thin organisms, but why not? So I'm going to switch over. That's my only phase contrast objective that I have. It's not in phase contrast yet, because I have to change the condenser here, and it's too dark because now I have to open the condenser, okay? And this is phase contrast. No, it's not dark field, it's a phase contrast. Yeah. So phase contrast converts differences of refractive index into bright field. Yeah. So yeah. So thinking about this, I think I should have added a little bit more methyl cellulose. X-ray look, yes. Yeah. So usually, I know that occasionally people contact me and say, I want to have a phase contrast microscope, and sure, fine. But normally for observing water samples like algae and whatever, usually they're much thicker, and therefore it doesn't look quite as nice. I just have to say, phase contrast is not, I mean, is good for some specimens. For this one, for example, it looks quite nice. But for other specimens, it's not really the ideal contrasting technique. Yeah. How do you adjust your DIC on your microscope? Do you have sliders? Have you tried sliding them in and out to change the contrast? Okay, I'm going to show you what I'm doing. So I'll go back again, let's say to this one over here. So what I have is I have a, yeah, so this is now 20 times, 20x. What I have is, how am I going to do that? I think I need to, you know what, I'm going to go to the desk view and I'm going to try to show it to you, boom, and I'm going to swing over the camera, but it's upside down. I hope you don't mind, okay? So basically what I have over here, I have to adjust this setting. You see this white dot over here, right? This has to match with the objective that I'm using, okay? Yeah, so that is that's a prism that's in there. So it must match. And the second thing that I can adjust, that's something that you can see up here, is I can change the color by rotating this knob here. And there is a slider. And I can completely take it out, and then basically it's completely removed. So to say I don't have DIC, or on the bottom, if I want to switch, it's easy just to remove this one because there's another prism down here, and and a polarizing setup and so on. Yeah, so I just basically flip it out, and then I have a regular bright field. So usually what I do is I just do this, to switch. Yeah. So I don't agree that bright field is natural colors. Natural colors is oblique illumination. Well, actually, oblique that's a little bit a question of definition, maybe. I mean, natural colors. What I meant is the microscope does not add any colors to the specimen. Yeah. Ultimately, you're always going to see some kind of refractive patterns as well, darker edges and so on, which are of course also not there in nature. So the microscope always will change, of course, something. Yeah. Well, let's go again to the scope view. So here we are again. Yeah. So that's these are basically the worms, and if you really want to see a lot of them, let's try that as well. Because let's try this. Let's try it again. Let's go back to the desk view. Yeah. So let's go in here because I've seen that many of them also collected on the bottom here. So I just want to try this, and let me double check also the time a little bit. And then I'm going to I want to reculture them, and then hoping that So I don't know. I have to go in right now a little bit deeper because many of them are on the bottom, and I don't know if this is actually going to work or not. But let's give it a try. I don't know. I don't know if there are more of them. Let's try maybe one over here as well. Sometimes they're clusters together. Yeah. I don't know. I'm just going to okay. Try it like this, and I'm going to add again a little bit of this methyl cellulose. Maybe a bit. I don't know. Maybe a bit more. I don't know. It's so thick, the methyl cellulose, that it doesn't always suck up into the pipette. Now I got some stuff up here as well. See, it doesn't even come out properly. No, it doesn't come out properly. So what I would probably do is the following. I'm going to try something different. You see, if you have problems taking up some of the liquid, then you might want to try the following. You take some scissors, okay, and you simply cut off the tip. Okay. And then let's try it again. Yes, this is actually better. So it's quite a lot more. Mix it together somehow carefully. So again, I think microscopy, like many other things, it's a lot of trial and error, and the more often you try, then the luckier you might be. Yeah. Cut the tip. Yes, I cut the tip. Yeah. Because sometimes it's like this. That I mean, you can do it. By the way, look, these disposable pipettes. Sometimes they're too thin, and sometimes I would like to take up some larger organisms like water fleas, small crustaceans, and so on. Then I also cut off the tip. Yeah. So simply to make the diameter larger. By cutting off the tip, you're not changing the amount of liquid that you're actually taking up. So this has nothing to do with that. So let's try it again. And I don't know. Let's give it a try. And I need to. Here we go. So let's remove those slides, and let's put them in here again. So. I think this was not a... they're moving faster. So maybe I did not get enough... Oh, here. Okay. Here there are more of them. Okay, that's a little bit more interesting maybe. Huh? Yeah. So you can actually see there are smaller ones, larger ones, and so on. Yeah. And again, in phase contrast, just because they're so fun to watch. Oh, yeah, you see, you see, it's already too thick a little bit, so it doesn't quite look as nice, I guess. Yeah. Yeah, I think maybe we just leave it, yeah, the way it was before. By the way, if you can also use the phase contrast setting, and now this here is dark field. So right now I'm using the phase contrast annulus, and this can be a little bit abused, also like a dark field filter, but it only works with the lowest magnification in my case. Yeah. You see, yeah, it's still kind of kind of fun to look at. I mean, it's one of the things that yeah. So let's close this again a little bit here. Yeah. Let me quickly have a look at some of the comments. Yeah. There are special micro pipettes to pipette viscous fluids. They have disposable tips with plungers. We use them in our lab to pipette detergents. Yeah, how much do they cost? I don't know. I can show you the pipettes that I'm using. I've not shown them. These are so-called microliter pipettes. And yeah, these are the cheapest ones I could get. But the ones that I used in lab many years ago, they were crazily expensive. One of them costed around, I don't know, 300 euros or dollars, several hundred euros. For one of those, I think I paid around 30, a tenth of it. They're not quite as good in the quality, right? It means because sometimes they do, I don't know, they do fall apart a little bit and you have to put them back together. But they work really. I think the reason why they're so expensive, the others, because they have been calibrated, yeah? And I don't know to what extent they are actually accurate, yeah? I mean, they have a precision of a tenth of a microliter, and they're good enough for my purposes, really. I've got two of those. This one is a maximum of 50 microliters, and this is a bigger one here, yeah? And yeah, you basically, what you do is by pushing this button here, you're kind of throwing off the tip. I can just hit to demonstrate this to you. If you don't know how this works, yeah, you have a tip, yeah, a disposable tip, plastic tip, right? And by taking up the fluid, you basically push this button here. Over here, you can adjust, by turning this, you can adjust. You know what, I'm just gonna show this to you here on the desk, yeah? Here, by turning this, you can actually, yeah, adjust the volume. So it looks like, and if you want to throw it off, you push this button here. When you push this button, then basically the tip flies off. Yeah, and this one was a cheaper one. That's why you press too hard, and sometimes the whole pipette falls apart. So yeah, but I think the reason why you can take it apart is also for cleaning purposes. Yeah, so just saying. Yeah, because you had a question. So that's basically the scope view, and yeah, so you see that this culture seems to be healthy, and I now want to subcultivate them, and then I would like to move on to the next topic, right? So how am I going to do that? I've organized myself somewhere, and I don't know where I put it. Here it is. Where is the desk view again? Here, a jar. And what I need to do now is I need to prepare the vinegar solution, and the recipe is 50% vinegar and 50% water. I have an idea. I'm a little bit concerned. Maybe I'm not going to do that yet. I'm just thinking. I want to see if the vinegar eels here, the nematodes, are able to survive concentrated vinegar. Why not try that? Because I'm kind of concerned maybe the reason why the other culture got moldy maybe is because I diluted it with water, and maybe the pH was already not right. Yeah. So then, yeah, let's try that. You know what? I'm going to try something very cruel now. I'm going to simply mix some of those vinegar eels here that I hope to be able to extract from. Let me see. I'll put it directly under the microscope to see if there are any there. So I'm just going to put this one in here. And without cover glass now, let's have a look. Yeah, plenty. Okay, so they're moving quickly, right? Why are they moving so quickly? Because I did not put a cover glass on top, so they have completely free movement. And now I'm going to try something. I'm now going to put directly some concentrated vinegar on top. I want to see if they're able to survive this. Because this helps me adjust the vinegar concentration. So I've got some concentrated vinegar here. Okay. And carefully I'm going to directly, you can actually see what I'm doing up there. I'm directly going to drop the concentrated vinegar directly on here. And I wonder if this kills them. I think it kills them. Yeah, they didn't like that. Let's have a look. Okay. Are they starting to move again? Did they just recover again? Uh-huh. Let me go bright field. Oh, okay. Yeah, let's try it again. They seem to be happy again. Maybe this was just a short pH shock. I still have a little bit of concentrated vinegar in here. So let's add another drop. Some air bubbles. Let's try it again. Let's add more. Let's mix a little bit. I'm pulling it in and out. Let's mix. It looks like maybe they have indeed. I don't know. Okay, they seem to be doing fine with the concentrated vinegar. But I think it looked almost a little bit like, well, this one seems to be dead. It looked a little bit like they stopped moving very briefly. Maybe it simply took them a couple of seconds to get used to the new pH. And if you get a strange result or an unexpected result in the sciences, what do you do? What do? You repeat the experiment. And that's what I'm going to do now. I want to see, because then that would be kind of interesting. So let's try it again. Okay, the desk view again. I'm going to remove now those here. And I'm going to, again, do the following. I'm going to take a, how do you say, here, new pipette, new slide. Where are my slides here? So let's have a look. Let's go in here. I don't know, maybe from the bottom. I think over here there are more of them. I don't know. So up, yeah, here it is on the very bottom. Yeah. So let's have a look. If again, I'm going to change to the microscope view. Yeah, okay, a lot of them here. I'm very happy. So, and now again, a drop of concentrated vinegar, because in the jar it was not concentrated, it was 50-50, right? So the pH is quite different. So I'm going to add now quite a bit of this concentrated vinegar here. Let's mix quickly. I could not repeat the experiment because they seem to be doing fine. I don't know why some of them stopped moving before. This one here in the middle is having some issues maybe. Yeah, try centrifuging them into a pellet. Yeah, that could be. So let's do the following again. Let's take add another drop because... Yeah, but this is actually important for me to know, because this means that I can actually go up with the vinegar concentration, and it doesn't hurt them. And then maybe if I go up with a vinegar concentration, then also I can prevent the growth of those other contaminants. They're doing fine. Yeah, Yet one more. Yet one more. Yup. Some of them move a little bit weird. Yeah. Yeah, so those small creatures are weird. Right now, I probably have bacteria in the diesel tank of my ship. I had engine problems, I thought, which turned out to be diesel bacteria. Not surprising me. Indeed, when I studied at university, they actually said that there are actually oil companies often have real issues with bacterial growth in oil and in gasoline. Yeah, because they're using the hydrocarbons as a food source, and this can actually cause the clogging of pipes and so on. Yeah, having bacteria growing in areas of hydrocarbon like diesel and so on, not is something that does not only not surprise me, but is actually something that has been documented. And I know that I've read and heard, and we've learned of this, that those oil refineries and so on, they actually have some issues with bacterial growth in the piping. Yeah. So that essentially it can actually inhibit the flow and clog the flow. Okay, so yeah, so this basically means I can go up quite a bit with the acid concentration, and let's do that. Okay, so let me quickly check a little bit also the time. 45 minutes now. So we now go back to the desk view, and I'm going to now use a little bit more vinegar than before. So this means maybe 2/3 of vinegar and 1/3 of water. Okay, so I need to get my water. The water is here. I always keep the water in a plastic bottle because I do not have running water where I am right now. This plastic bottle is deliberately black to prevent light from entering it. Why? Why that? Because otherwise you have the problem of algae growing inside the water bottle. So that's the issue. So for example, I can show you over here. This one is a clear water bottle. It contains alcohol, so no algae growth. But for water, it's a problem. So let's, I don't know, add a little bit. I'm just going to, that's not cool. It's too slow. Let's add a little bit of water here. And I'm going to add maybe twice as much vinegar now. I'm using this cheapest apple vinegar that I could get, just estimating the whole thing. So I'm just about up to here. That's a lot. So plus you can of course add some apples, a small piece of apple. I'm not gonna do that. I'm going, because I was also successful in the past. This is just sugar, regular table sugar. It's part of the recipe. That's also a food source. And then I don't know, I should have mixed it actually. I didn't. Yeah, it's gonna dissolve on its own. And yeah, I think I'm just going to leave it as it is. And I'm going to then add simply a small amount of those vinegar eels. I'm going to use my larger pipette now because I simply want to grab a larger volume. And I'm simply going to do the following. You just go in here, you grab yourself a lot of this stuff in here. You can see it's quite cloudy. You put it in here, and that's it. And now I'm just going to wait for, I don't know, a couple of weeks maybe. And of course I'll report back. Yeah, so we're gonna see whether they actually will survive or not. Let's put a lid on top here. Yeah, and this one I'm just, I should actually also put a lid on top on this one here, but I'll just put it on the side now. Okay. So, how much sugar exactly? I tell you the following. The recipe, I mean, there are different types of recipes. The official recipe that I researched, that you can Google this because a lot of people are actually growing those vinegar eels, those nematodes, because for fish food, they always say around 50% vinegar plus 50%. I think 50%, 4% vinegar plus 50% water plus a spoonful of sugar. And then that should be fine. And some suggest that you also add a tiny piece of apple for additional nutrients. Honestly, I don't think that this is so important because the sugar concentration and the vinegar concentration changes anyway as those worms grow. Yeah. So that's the thing. It's not gonna stay constant anyway. So that is basically the one thing that I just wanted to show you. So I think we've played around enough with those worms here, because I do have to mark the new jar. Where's my pen? Okay, let's mark the new jar. What should you put on here? Okay, let me show desk view. Yeah, because sometimes I forget to label it, and then so I know it's vinegar eels, but today is what, October 4th. October 4th, yeah, and that's gonna be fine, yeah. And we're going to see. Actually, in order to prevent this from being wiped off, you can put some tape over it, yeah. So I know what this is because I only have those, yeah. The only one, this jar. So let me quickly see. Yeah, so let me quickly see what else I would like to do. So I'm going to do very quickly, because I also want to know that, is I'm going to now put some of the one Hyalozoa worm under the microscope. They're here. And okay. I need to put on my reading glasses now. So, and I need to, there are plenty of them in here. How am I going to do that now? I need to take again a pipette, and this is now a little bit a question of luck whether I'm able to catch one of them. You cannot see that. See here we are. I don't know. Yeah, that's a little bit not quite in focus, unfortunately. Yeah. You see some of those dots moving around. These are still some Daphnia, right? But here on the side you have plenty of these worms. Sometimes when you suck them in, they break. So I got this one. So maybe there's. I hope that there are at least one or two in here. So I put them on here. Okay. Sometimes you can actually see them if you put them on a darker surface. This is gonna be my dark surface. There's a reason why I bought those mice for demonstration purposes. Yep. Can you see those? Can you see this white stuff here? Right. That's a worm. Right. So what I'm going to do there for now is I'm going to remove the excess liquid, because I want to have very little. Let's see. It's not moving. Why not? Maybe I accidentally cut it apart when I sucked it in. Those mice here that I bought here are actually because I'm working on another video about Robert Koch, who did some mouse experiments back in the 19th century, and these mice here are the models, so to say. So scope view. Let me see if. Nah. Nothing. Okay, I gotta try again. No worm. This is not a worm. This is some kind of other stuff. Okay, I gotta try it again. This was not successful. Yeah, doesn't matter. There's so many of them in here. It shouldn't be a problem catching one of them. try it now. I think. So, scope view, where is it? Yeah, here. Ah, yeah, here I got how many? 1, 2, 3, 4, 5. Here they are. Okay. Yeah, and I will tell you. Yeah. Shall we try something? I would like to do the following. I would like to add some methyl cellulose, and I don't know if I have time today. I've got some caffeine here. And this caffeine, if you add, I tried that yesterday, if you add caffeine to those worms here, they do not go faster, but it actually paralyzes them. But they're still able to move because of the cilia that they have. Yeah, we're not going to do that now because I'm more interested right now in the how do you call this, in slowing down their movement. Because I really want to limit their movement so that maybe we can actually also see the tiny cilia. And I need to go back to the desk view. And here it is again. And now I'm going to add again some of this methyl cellulose. It's quite a bit. You do have to slightly mix it. If you don't do that, then there is going to be a region on the slide that's still very thick and the other region is completely thin, and that's also not good. And I hope that I didn't... Could of course be that some of them are still in here now, but we're just going to try. So, no cover glass. And the scope view here. Let's see if we're able to find. See there. Here they are. Ah, yeah. They are basically a little bit. How do you say? Difficult to see because of the methyl cellulose. So I'm taking a toothpick and I try to pop some of those bubbles here. Okay. Yep. And we can now try to go a little bit up with the magnification. Yeah, it's actually much better. Well, they're still moving fairly quickly. Yeah. In reflected light, I don't have unfortunately a flashlight with me right now. But what I want to do now is, in any case, I'm going to drop quickly a cover glass on here. Let's see if this has... Here we go. And now we can really try to... Yeah? Polychaeta, aha. I thought they're oligochaeta, those worms here. Interesting. Yeah. Yeah, they're still moving fairly quickly. However, what I can try to show you is, and that's also something that I was able to see, those bristles actually are able to move, and you can actually sometimes see the muscles that move the bristles back and forth. Yeah. Yeah, but still, yeah. Still a little bit difficult to see, but yeah, but a little bit maybe here at the head, on the surface, you might be able to see that there are these tiny little hairs that are beating and moving. Yeah, but it's always moving in and out of focus. But occasionally you can actually see them here. Let me turn on the arrow so that if I am able to see it on the surface. Let me. Yeah, you can actually see those tiny cilia here, yeah, here that are kind of moving as well, and they're generating this water stream so that they're able to feed. Yeah, where do they grow? How can I hunt them? Oh, they're easy. I mean, they're almost unavoidable. If you get yourself some kind of a from the pond, If you get from the pond some sludge, yeah, with the algae, I mean, I almost always find them in there. I was kind of lucky with this one because they reproduce so quickly, and last time, and I don't know if you're able to see this here, but they reproduce also asexually very, very quickly by dividing in half. Yeah? I don't see that over here, but sometimes maybe in last week's video, I actually was able to find a few of them that actually were growing a new head right in the middle, and then they're going to separate after some time. Yeah. And yeah. So one thing that I've also done in the past is I've been feeding them some food, of course. Then you can actually see how they're eating the food. Yeah. So let me quickly do the following. I want to quickly go again back to the chat here. Yeah. Why does the caffeine paralyze them? I don't know. I can actually show you how this actually looks like. I mean, there are some school experiments that you can do when you add a little bit of caffeine to some of these water fleas, water crustaceans, and it changes the rate of the heart rate. I've also seen that some of the water fleas were jumping around much faster after you've added caffeine. And I've seen yesterday, at least, when I added a little bit of caffeine to those worms, they actually started to basically become paralyzed. So only the head moved and the rest of the body was simply dragging behind. You know what? Let's try it and just show you. Because after all, I'm a crazy scientist. Why not do a couple of experiments? So I'm putting them away now, and I need a fresh, yeah. And this here is now simply a caffeine tablet, and caffeine, I have to dissolve it in water. Caffeine does not dissolve well in water. Yep. So that's yeah, one thing. It does not dissolve well in water, but I hope that this is enough. Let's wait a little bit until it breaks down again. This was simply a tablet that of pure caffeine. I think it's not just caffeine. Maybe they also added some kind of a filling substance. So this is going to look a little bit ugly with all of those particles floating around. Just this, yeah. But let's try this as well. And then I promised you very quickly also to, what time is it? I promised you also very quickly to show you how to make this spore slide. So let's again try to grab one or two of these worms. I don't know if I got one or not. No, I didn't. So let's give it another try. Yes, I think I now did. So let's put it again under the microscope to check. So let's see if there are somewhere. Here it is, right? Here's one of them. It's the second one, I don't know. Let's go down with the magnification. I mean, one is enough, really. That's the edge of the water drop. Is this the same one? No, there's a second one. There are two of them here. Okay? Yeah, here they are. So what I'm going to do now is I'm going to add a little bit of this caffeine. And then let's see if I'm able to reproduce this little experiment from yesterday. You can actually now see that the kind of the whole body is kind of, yeah, moving. So they're kind of contracting and expanding, right? So I'm going to add now a little bit of this caffeine. You can actually see what I'm doing. I'm just going in here and taking out a small sample. And now when I add this, there are going to be lots of particles floating around, right, from the tablet. So it will not look quite as nice. Okay, just saying. Let's add this here right directly on the top here. Yeah, see all of these particles here that's kind of from the tablet. And then it takes a minute or so until it starts to work. Where's the other one? Where's the larger worm here? Here's one, here's the other one. And then let's see. This actually also has the advantage that we're now able to see a little bit the flow of the water at the head. Here's the other one. Okay. Yeah, it's still contracting. I don't know if I'm able to repeat the experiment. At least yesterday, it is something that kind of surprised me a little bit. That then all of a sudden the worm was completely extended, and only the head was kind of moving. The cilia were moving and pulling the rest of the worm along. But right now, you still can see those peristaltic movements of yeah. Let's see if this will change or not. Okay. So why does caffeine paralyze them? I don't know. Evidently, it does seem to have some kind of an impact, and we're going to see. I don't want to promise too much. We're going to see whether I'm able to reproduce the experiment. Yeah. It's still there's still this peristaltic movement. Okay. Let's see. If it doesn't work, then well, I was not able to repeat the experiment. Yeah, but this one already starts to behave a little bit weird. Okay. Yeah, maybe somebody can do a little bit of research. According to my knowledge, those Ailozola worms are of course segmented worms, obviously, right? And they apparently belong to the oligoheat worms. So those worms that don't have too many bristles. But you see, oh yeah, something's happening here already. Yeah. What about the other one? Where's the other one? This one still seems to be more or less. Yep. Yep, so that's basically also what I was able to see yesterday. Yeah, the peristaltic movements seem to, however, at the head, the cilia continue to beat. Now you can actually see the movement of the water, and this, of course, indicates that this worm has two ways of movement. The cilia at the head, which of course are not controlled because they're on a cellular level, they are not controlled by nerves, and evidently the rest of the body, which is controlled by nerves. Yeah. So and now when it's moving around like this and swimming around, then kind of the cilia at the head kind of pull the rest of the worm, which is now does not do this peristaltic movement anymore, or at least not as much. Thank you very much. Okay, there are a couple of questions again, which mostly I answered already, but we're going to have a look. Yeah. So this is actually also something that completely extended, and you see also the intestine on the inside, it doesn't move anymore. Here's the other worm. This one still seems to be moving a little bit, so maybe it was not in the area where there was a lot of this caffeine. Yep. I don't know if it's possible to reverse this or not. So let me quickly have a look again at some of your... Let me have a look here. With high doses of caffeine, neurons are so strongly stimulated that they stop conducting impulses properly. Aha. Yeah. Okay. So according to the wiki, they are either attributed to oligoheats or polyheats. Interesting. Yeah. This one still is moving here. Look, this one over here still seems to be wiggling around, and the other one over here. And I wonder, I don't know, I wonder if after a couple of minutes or so, they're getting used to the caffeine that they actually start to behave normally again. That's something I didn't try. Yeah, question is, just woke up, what's in this stream? Well, I for the first half of this live stream, I talked, for those of you who just joined, I talked a lot of about those nematode worms. And now this is basically Iolosoma. Now it contracted. Iolosoma, which basically has been treated with caffeine. And apparently the caffeine starts to paralyze it a little bit. But I'm just wondering if after a couple of minutes it starts to move again, as it maybe, look, peristaltic movements start again. Maybe it's getting habituated to the caffeine. I don't know. Huh? So that's kind of where it's developing some tolerance to it. Yeah. We're going to see. Yeah. In any case, you see that the head here, the cilia of the head continue to beat. Yeah. I found that the caffeine inhibits the acetylcholinesterase enzyme, which usually breaks down the neurotransmitter acetylcholine, in humans, and therefore it stimulates neural activity. But also in humans, at least, I guess maybe also here, if nerve cells are overstimulated, then they will down-regulate again. Yeah, so, and you see, occasionally we do now have again some peristaltic movement, and maybe that's, an indicator that the worm maybe, or the nervous system of the worm starts to become tolerant. I don't know. Yeah. So let's 1 hour 11 minutes already. What I would like to do now is, simply because I promised you, I need to quickly, somebody asked, how do you prepare spores for microscopy? Let me quickly get some mounting medium. So let me see, I have some U-Pearl mounting medium here. Where did I put the other mounting medium? I forgot. Elmer's PVA, here it is. Here it is, here it is, here it is. Where's the worm? Here it is. Yeah, completely extended. Huh. Yeah, completely extended now. And now, and now that's actually the thing that I've also seen yesterday. It's gliding along, and now just being pulled by the cilia at its head. Yeah. Interesting. Yep, this one down here. Yeah, slowly also starts to. Okay, so you know what? Because it's already over an hour. Yeah, if you want to know how to make mold slides, you do the following. You grab yourself a slide, a microscope slide. Okay, and where is it? Yeah, and it's fairly easy. This is a very, very easy one. I've got some spores here. Okay, you do the following. You grab yourself some glue, some water-based glue. The one glue that I'm using is this one over here, but you can use maybe also other water-based glues. I diluted it down a little bit and put it in here. So this is just a regular glue for, it's also used by children. You try to open the bottle after it's been, and you put a small amount of the glue over here on the microscope slide. You do the following. You go with a toothpick in here to pick up a little bit of the glue to collect. You spread it. Okay, and what you probably want to do is if you're using a mushroom, you put a drop of glue or mounting medium of your choice on here. You put the mushroom cap over it and you knock on it to knock out the spores. It's a little bit fuzzy, I see. Okay, I think should be enough. Cover glass goes on top and you just wait for the glue to dry. Just very quickly now, simply to proof of concept. And we go into the scope, microscope again. Yeah, and you see the spores are still floating around. Kind of a little bit, kind of a bit small. Let's go up. Yeah, and if you see some longer cells, actually this is not a good one. I've actually probably not caught a lot of them. Here, it's better. Those tiny dots. Yeah. Let's go up. Here, that's better now. Okay. Those oval structures that you've got here, okay, are the spores. Yeah. So, yeah, so that's a little bit a very quick, yeah. Yeah, there is that's a point. Yes, please. Because there's always every day I'm getting a email or two for people who are not who can't get into the form. You have to send me an email. I have to unlock you manually. The reason is the following: that in the past we had so many spam registrations. It really was horrible. And then I actually have increased the safety by writing a comment. You have to write a comment. Why do you want to join the forum? Hoping that and you had to answer some questions and so on. Hoping that this way I'm able to filter out bots. And but these days with AI, it's also possible to fill it out, right? So this is a problem that I've had. So what I had to resort back to is a little bit the how do you say this, manual unlocking. So if you send me an email. And currently I do have the problem as well, a technical problem with the forum, that I'm getting a page not found error every time when people want to change the password. That's I'm gonna make an update, and I hope that this will also resolve that issue. Yeah. So this is indeed something that I how do you say, I'm still working on. Yeah. So what else I'm gonna do now? So this was very quickly, so a very easy way of doing the how do you say the a permanent mount. And what I was not able to do today, but then I'm going to leave it maybe to next week, is I was not able, but there's no hurry, I was not able to show you those other fungi over here. But this is actually something that would take a little bit more time. Yeah, and I would like to quickly go through the questions here, if there's anything that I did not answer yet. Yeah, that was something of permanent slides with the fungus, at least the spores. And if you actually want to look at the cells of the fungus, then of course you need to kind of also cut them, slice them. And it depends a little bit if you're looking at mold or if you're looking actually at the fruiting body of a fungus. Yeah, so that's what I just showed. I have a cam question. So that is use a Canon camera using a 2x adapter. Well, the thing is the following. Actually, what you have to do, if it's kind of very often it's like this, that if you want to use a camera with a large sensor, then you have to use a magnification sometimes which is actually lower than because it has to make it wider, the image. I think I'm gonna make a separate video or live stream. Yeah, I don't know which fungus it could be. Okay, camera setups, we talked about this. I have a Sony, again a camera question, a Sony A6000 fitted on the third tube of the Swift directly on the C-mount without any glass, but photos are not sharp, even a stacking series. Any suggestions for a specific adapter? Generally, I have to say the following. That's difficult. Generally, it's like this. If you do not have any intermediate optics, the image should be quite sharp. I'd have to look at the pictures. And yes, it is complicated. That's essentially one of the things that I've found out. That even if you have an adapter, it's not a guarantee that it's going to work well. So I've got a whole bunch of adapters, and some of them are horrible. really. And then all of a sudden, some of them were that were horrible works pretty well with another camera. So there's lots of trial and error going on here. Yeah. So there are special micropipettes to pipette viscous fluids. Okay. Yeah, I don't know if I'm able. Yeah, I can try to centrifuge the worms. But the question that I have is, yeah, I have to work pretty fast because as soon as I stop centrifuging, they're going to start swimming around again. Yeah. What exactly is vinegar? Vinegar is a fermented product. It contains acidic acid, around 4%. Yeah, basically it's formed by certain bacteria can actually form also vinegar commercially out of out of apple juice, for example. The apple juice is contains sugars, and then when it's fermented, it's converted to an acid. And this actually means that some juices, when they start to become bad, they will turn acidic because certain bacteria convert the sugar not only into alcohol first, but then from the alcohol is then further basically oxidized into an acid. Yeah, so it's a multiple step process. Sugar is converted to alcohol. Alcohol is converted to acetic acid, for example, and then you have a vinegar. Yeah. So that is the thing here. So yeah, there are lots of comments here. I'm quickly going to go through some of the comments here. So there seems to be quite a little bit of interest concerning camera setups here. Okay. You know what I'm gonna try is, yeah, I did mention it actually in the form in the how do you say in the rules that you have to write me an email. But what I might actually do concerning the format, what I might actually do maybe, I might actually make a video which you have to watch before signing up, and then maybe I can tell you a password inside the video. That would be not a password, but some kind of a, how do you say, an answer to a question, and then I actually know that you're a human being because you've watched the video. That might actually be a possibility for the forum. That's I might do that, because then I also save myself quite a bit of time of having to manually unlock each person. Yeah. So that's a little bit the problem that I've been having, because a couple of years ago, the situation with the Microscopy forum got so bad that people actually started unsubscribing to it because they could not, because I was not checking it for a few days, and then it was full with spam, right? And content that's not good. Yeah. So that's one of the reasons why I then said I have to do something. It took me quite a bit of time to delete all of those spam comments in the forum. Yeah. So yeah, okay. I'm just reading here. Yeah. So let me quickly see if there's still something going on here with the worms, and otherwise I'm going to call it a day for today. Here they are from the still alive, and you see that's the nice thing about the methyl cellulose. Yeah, it does not seem to impact. It doesn't modify the osmotic pressure of the liquid, and therefore the worms, they do not dry out. Yeah, for example, if you try it with glycerin, which is also a very thick and viscous liquid, glycerin is going to kill them. Yeah, because glycerin will pull out all of the water from those worms. And that's not gonna work. But this methyl cellulose actually seems to be working quite nicely. Yeah, and then let's go up yet more. And now you can actually, and yet more. It's now 60 times objective. I need to open it here a little bit to get it more light. At this magnification, without the methyl cellulose, it would be almost impossible to observe because they're moving so quickly. Yeah, up. Fascinating. I mean, those creatures fascinate me. And you know what? I'm working again. Also, I don't know. You might have. I'm working again on the video, but you know that I have not been uploading a lot to this channel because I was kind of busy the last week. But I'm planning a video on how we humans are similar to those worms. I'm not kidding. It's one of the most difficult concepts to grasp for non-biologists, and that is that all living things are very similar on a very basic level. So the cellular structure, for example, is very similar, and so on. The genetic code is the same, almost the same in all living things. And basically there are so many similarities on a very basic level, and the deeper that you zoom in, the more similar the living things become. That's, I think, one of the most difficult things to grasp, because we're looking at the similarity of organisms and not at the differences. I'm getting a little bit philosophical here, but that's actually one of the reasons why I'm also going to use those worms, yeah, as an illustration that on a very basic level we're quite similar to those worms here. Yeah. So let me quickly fly through the comments here. Yeah. I mean, it's interesting. I didn't know that some of you are actually watching the whole thing on television. I mean, yeah. I hope that the resolution is fine. Yeah. So, and I think it's smarter to let the password appear on the screen in the video. Text-to-speech systems are quite advanced these days. Yeah, that's also a point. Yeah. Makes it maybe also easier to change the password by just changing the text. And then I will be asking a question, or I'll be asking for the password, and then you just type that in, and then you should be able to sign up, basically on your own, and it's not necessary anymore for me to actually do the unlocking. Yeah. So I think I'm going to really call it quits today. I hope that I answered most of your questions. Yeah. What event was that? Public display? Let me see. I think somebody was watching it on television or so. Let me see. Or was this really on a public... Thanks for your enthusiasm. Thanks for your tutoring. A recent public display with a big TV, a great success. What do you mean? Can you please tell me a little bit more? Dave Lewis, was this kind of was this stream somewhere publicly shown somewhere? That would be kind of interesting. Yeah. I know that once I got occasionally get some notifications that some of my videos are actually shown also at the university, concerning slide preparations and so on. Yeah. But a public display would have been the first. Yeah. Yeah. You can even see the viscosity around the worms. Yes. Yeah. Sometimes you see a little bit the differences in refractive index. Yeah. I wonder if you can increase the reproduction of a certain organism in your sample. Of course, you can add more food or oxygen. Also, could there be too much oxygen in a sample? Generally, too much oxygen, I would not think that this is an issue. Okay. Yeah. But I know that sometimes if the oxygen actually is highly oxidizing, obviously, so for certain bacteria, they will die if you have too much oxygen because they're anaerobic. But normally for animals, I don't think so. Yeah, because the oxygen concentration is not going to be greater than what you have normally in any case. Yeah. And also, if you, for example, if you put algae or plants into the water sample, then they will produce a lot of oxygen by photosynthesis, and this also does not harm those microorganisms and those animals here. Yeah. So, folks, I think I'm going to Dave Lewis. We had a local natural history show. Okay, that's nice. Okay, and I'm gonna call it a quits for today, and. Yeah, I'm going to think about maybe we're going to look at cheese next week, and maybe also add some ways of how you can connect a camera to a microscope. Okay, yeah, I'll wish you all the best, and happy microbe hunting as always. And I'll be uploading again in the next couple of days a couple of more videos. Do also check out my other channel that I have, where I'm now going to upload more explainer videos, slightly more talking head type, but yeah. Plenty of stuff to keep you busy with your microscope. Okay, all the best, and happy microbe hunting, and bye-bye.